Hi class! So this week we will continue to study about the biological molecules, which are carbohydrates, lipid, proteins, and also nucleic acids. However, we already cover subtopic of carbohydrate last week. So today we will continue to study about another three biological molecules, which are the lipids, proteins, and also nucleic acids. So the first one is lipids. So lipid is a biological molecule in which the structure is mainly consists of carbon, hydrogen, similar with the carbohydrates. However, they attach with a few functional group that containing the oxygen. So due to this functional group that containing the oxygen, it causes the lipid to tend to be hydrophobic molecules, meaning to say they are insoluble in water. Therefore, the general properties of lipids are they do not have polymer, so meaning to say lipid does not come from a monomer, such as the carbohydrates. Okay, they already have their own structure that cannot make up any polymer from the monomer. And also lipids are greasy or oily compounds. Okay, if they are solid at room temperature, if they are in a solid state, they the characteristic usually very greasy. However, if they are in a liquid state, usually the lipids are known as the oily compounds. And as I mentioned earlier, the lipid is hydrophobic, meaning to say that they are insoluble in water. However, they are soluble in organic solvent such as the alcohol, benzene, and also chloroform. So there are three major classes of lipids that you have to remember. The first one is triacylglycerols, or also known as fat, or triglyceride. Okay, so triacylglycerol or triglyceride or the fats, okay, can be divided into two types, which are the fats and also oils, in which the triacylglycerol is made up from one molecule of glycerol fused together, bonded together with three molecules of fatty acid. In the second group, or the second class of fats is phospholipids. Sorry, there are four classes of lipid. The second one is phospholipid, the one that make up the plasma membrane. And then we have steroids such as cholesterol and also the testosterone hormone that you can find in male. And then also wax, okay, the one that very important substance that make up a layer on the leaf so that it can reduce the loss of the water from the leaf of the plant. So lipids, similar with carbohydrates, protein, and nucleic acids, okay, even though they are not a polymer, however, they can form a very large molecules from a small molecules, okay, through the process of condensations or dehydration, meaning to say by removing the water to form a bond between the small molecule so that it can produce a large molecules. Okay, and normally the smaller molecules that make up the lipids are glycerols and also fatty acids. So there are many importance of lipids. Okay, the first one is that they highly reserve energy storage, meaning to say they can store a lot of energy, or they can reserve a lot of energy compared to carbohydrates when our body need a lot of energy to be due to our high metabolism. And the second one is it is important for as a cushion, okay, for the internal organs or tissue. Therefore, when there is a sudden shock, okay, the lipids, okay, can absorb the shock. Therefore, to avoid from the shock damaging or the pressure to damaging our internal important organ. And then it also as a protective layer, as the wax layer on the plants, to reduce the rate of transpiration or the loss of water from the leaves. Okay, as the insulator, as the components of the plasma membranes, okay, such as the phospholipids, it's important for our immune system as the emulsifier, and also important to transport the fat soluble vitamins. Now let's look on the first group of lipid, which is triglyceride or fats or triacylglycerol. Okay, so fats or triacylglyceride are constructed from two types of smaller molecules, which are glycerols and also fatty acids. So what is glycerols? Glycerol is it is a molecules that have a 3-carbon skeleton. So meaning to say it have 3 carbon that make up the backbone of the glycerol, and it have a hydroxyl group that attach to the glycerol. Okay, here 1, 2, 3, it have 3 carbon as the backbone, and actually here we have OH group, which is the hydroxyl group OH. However, OH, the H already being removed due to the condensation process, the loss of the water. Meanwhile, the fatty acid molecule is a very long unbranched hydrocarbon chain. As you can see here, a very long, this is fatty acid, unbranched hydrocarbon chain, and then they have here carboxyl group that attach at the end of the fatty acid. Actually COOH, however, H here also and another oxygen have been removed due to the condensation process. So the formula of triglyceride or triacylglycerol is 1 glycerols bonded with 3 fatty acids through the process of condensation. Okay, as you can see here, 1 glycerol bonded together with 3 molecules of fatty acid, thus make up triacylglycerol. Okay, and the bond that form between the glycerol and these 3 molecules of fatty acid is known as a ester linkage. Okay, so for carbohydrate, the bond name as a glycosidic linkage. Meanwhile, for the lipids or the fats, the name of the bond that formed that formed by the condensation process is known as the ester linkage. Okay, so as I mentioned earlier, ester linkage is created by the triacylglycerol through 3 series of condensation process. Here they will remove H2O, okay, thus forming 1 ester linkage, and then another condensation removing H2O, producing the second ester linkage, and the third condensation removing H2O, producing this one the second, the third ester linkage. Okay, so triglyceride fats or triacylglycerols, okay, if they are solid in at room temperature, they are known as fats. Okay, if the fat or the triacylglyceride or triacylglycerol are liquid at room temperature, they are known or called as oil. Okay, and the fats or the oils are very important for the storage of the energy as the basic components of the natural fats and for the insulations in human and also animal body. Now let's look on the first type of triacylglyceride, which is saturated fatty acids. Okay, we have 2 types. We have saturated fatty acid or saturated fat, and the second one is unsaturated fat. So the first one is saturated fats or saturated fatty acid. Saturated fatty acids can be found mostly in animal fat, okay, and they are solid at room temperature, and they help to increase the cholesterol, okay, content in the blood. Examples of saturated fatty acid are butter and also fat. And also, as you can see here, this one is glycerol, this one is the fatty acid. The fatty acid of saturated fatty acid are a straight change, and they have no carbon-carbon double backbone here. There is no double backbone between the carbon backbone, okay? Therefore, the number of the hydrogen that attach to this carbon is at a maximum number. Compared to unsaturated fatty acid, as you can see over here, okay, unsaturated fatty acid, they normally can be found in the plant or the fish fat, which is, we call it as the plant oil or the fish oil, okay, such as the olive oil and also the fish oil. Therefore, since they are oil, therefore they are liquid at room temperature, and these are good for our health because it helps to decrease the cholesterol in blood, okay, and they have, as you can see here, between the carbon and another carbon in the backbone, they have either one or more than a double backbone, okay, a double bond. This double bond cause the structure of the fatty acid to bend like this, meaning to say have a kick, okay, a king at the double backbone, at the double bond, okay. Therefore, the number, due to these double bonds, okay, therefore the number of the hydrogen that attach to this carbon is being reduced, okay. Therefore, the differences between the saturated fatty acid and unsaturated fatty acid can be differentiated in two obvious differences, which is we can see through the chemical structure or the physical structure. For the physical structure, of course, saturated fat are solid at room temperature, so they are being called as fats. Meanwhile, unsaturated fatty acid, they are liquid at room temperature. Sometimes they are being called as the oils, okay. And for the chemical structure, saturated fatty acid, remember, they do not have a double backbone at that fatty acid. Therefore, they have a maximum number of hydrogen atoms that attach to the carbon backbone of the fatty acids. Meanwhile, for unsaturated fats, as you can see over here, they have either one or more double bonds, okay, between the carbon molecules, between the carbon atom of the fatty acid over here. So therefore, it causes a lower number of hydrogen atom that attach to the fats, okay. The next group is phospholipids. Phospholipid structure is that they have two fatty acids, okay. As you can see over here, they have two fatty acids, not three fatty acids. They have two fatty acids that attach to the glycerol, okay. Almost similar with the triacylglycerol. However, they do have a phosphocholine head over here that attach to the glycerol, okay. So therefore, phospholipid is very special because they are amphipathic. They have a characteristic of amphipathic. What is amphipathic? Amphipathic means amphipathic is that where it have a side that hydrophobic, meanwhile the other side is hydrophilic. So as you can see here, the hydrophobic, the fatty acid here, the two molecules of fatty acids, okay, make up the hydrophobic tails, meaning to say this part are insoluble in water. However, okay, this part, okay, the head, the phosphate group, okay, that attach the phosphocholine head that attach to the glycerol is a hydrophilic. So meaning to say, phospholipid has two characteristics, hydrophilic and also hydrophobic, which is later known as amphipathic. Okay, so the phospholipids are arranged in a bilayer. As you can see here, this is the structure of the phospholipid bilayer that you learn later in the plasma membrane. This is the first layer and this one is the second layer, in which the hydrophobic head here, the phosphocholine head, are at the outside that will contact with the aqueous solution over here. Meanwhile, the two fatty acids that make up the hydrophobic tails of the phospholipids are buried inside at the inner parts, okay, because they want to avoid from exposing themselves to the aqueous solution, okay, and they are the major components of the membranes. Therefore, what is the importance of phospholipid as the basic component of the cell membrane and also as a boundary, okay, as the boundary or the barrier between the inner part of the cells and also the external environment of the cell. Next group of lipids are steroids. So steroids have carbon skeletons, okay, that consisting of a few string over here, as you can see, and different steroids have a different functional group that attach to its rings, okay, and the two common types of steroids are cholesterols and also testosterone. Okay, cholesterol important molecules in our body. However, the high level of cholesterol also can cause a bad effect to our body, that if the content is very high in the blood, and it will later contribute to cardiovascular disease. And what is the importance of the two types of steroid? The first one is cholesterol. Okay, the major components of the myelin. Myelin can be found in the neuron cell. Okay, and then major component for animal cell membrane and also as the precursor for the synthesis of other steroids. Meanwhile, for testosterone, okay, testosterone is actually the male sex hormone, in which it helps to responsible to stimulate the formations of the sperm inside the male reproductive organ, and then it also to promote the male duct system in the fetus, and of course it important for the secondary characteristic in male, such as the facial hair growth at your armpit, your beard, okay, and your mustache, okay. However, steroids, okay, steroid cannot be overused, because the overuse of the steroid will give a bad effect to your body. It can cause cancer, it can cause cardiovascular disease, and also it can cause the renal failure in your kidney. Okay. Next type of the next type of lipids are wax. So wax is the most simplest lipid, the type of lipid that the most simplest one, the most simple one, in which it is an ester that in which they have a long chain alcohol that attach to one molecules of fatty acid. Okay, remember the first one, triacylglycerol, have 3 fatty acids attached to glycerol, and And then the second one you learned just now is that we have phospholipid. Phospholipid were two fatty acids attached to phosphocholine head. And then you also learn about the steroids. And then now is wax. Wax structure is one fatty acid attached to a long chain of alcohol, which is the OH groups. Okay, and wax, of course, they are insoluble in water and very difficult to hydrolyze, and they can be found as the protective layer in the leaves, stem, and the fruits, animal fur, so that to reduce the loss of the water from the surface or from the skins of the animal fur and also from the leaves. Okay, examples of waxes, we have carnauba wax that used for the floor and also automobile wax. We have beeswax that importance for honey and eggs, and also parmaceti wax that you can find in the fats of the sperm whale. Okay, then next we have prostaglandins. Prostaglandins also a group of lipid compound that made up from only fatty acids. Okay, they only made up from fatty acids. And the most common types or examples of prostaglandin, we have prostaglandin E2 or known as PGE2, or prostaglandin F2 or known as PGF2. And the function is prostaglandin is for the maintenance of the tissue because they make up the lining of our stomach, and also it helps to help in the blood clotting in our body and control the blood pressure. Okay, prostaglandin usually the name of the prostaglandin usually using the prefix of PG. PG means prostaglandins. Last but not least is that we have sphingolipids. Sphingolipids is also one class of the lipids that they have a polar head, okay, group, and two non-polar tails, quite similar with the phospholipids. However, okay, it's quite similar with the phospholipids. However, the core of the sphingolipids is an amino alcohol that known as the sphingosine. So as you can see over here, the head of the sphingolipids is made up from the sphingosine. Okay, different with phospholipid where the head is phosphocholine head. Meanwhile, for sphingolipid, the head is the sphingosine's head. Okay, and the functions of the sphingosine is for sphingolipids for the signal transmissions in the neuron cells, and also to help in the cell recognition from between cell and cells. Example of sphingolipids are ceramides, okay, that can be found normally within the cell membranes of the cells. Next biological molecules is protein. Protein comes from the Greek word that means primary. So protein composed of carbons, hydrogen, oxygens, nitrogens, and sometimes contains sulfur, phosphorus, and iron. And the protein, similar with carbohydrates, they have monomer, and also they have a polymer. Okay, the monomer state, which is the building block of the protein, is known as the amino acid. And the long chain of the amino acid, which will produce the polymer of the protein, which known as the polypeptides. So the chain of this polypeptide will be either folded, okay, and also coiled in the structure, and thus form a specific structure that known as the protein. Okay. So amino acid structure, okay, the monomer of the protein, they have four important components that attach to its alpha carbon or the central carbon. The first one is that they contain a basic amino acid NH2 amino group that attach to the carbon, and then they also consist of carboxyl group COOH that attach to also to the central carbon, and then they have a single hydrogen atom that attach to the alpha carbon, and also the R group. The R group is also known as the side chain. So this R group, okay, is only abbreviation for the any group that attach to this carbon, alpha carbon, okay. And the simplest type of amino acid is actually glycine, because the R group of the glycine is actually a single hydrogen atom. So the structure will be amino group, carboxyl group, hydrogen, and another hydrogen here is the R group. And the R groups, okay, is differ from one amino acid to its amino acid. And actually this R group is the one that will responsible to give the specific properties to the amino acid. Okay, and some of the amino acids are hydrophobic, some of them are hydrophilic, some of them are also acidic, and also some of them are basic. Okay, and they are actually 20 amino acids that you have to know that normally can be found in the protein. 6 amino acids are non-polar out from 20. A non-polar, which is we have glycine amino acid, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and also proline. Meanwhile, 6 out of 20 amino acids are polar, meaning to say they are soluble in water, such as serine, threonine, cysteine, tyrosine, asparagine, and glutamine. And then we also have 5 amino acids that have charge, which is 2 of them are acidic, such as aspartate and also glutamate, and also basic in characteristic, such as the lysine, arginine, and also histidine. So amino acid, some of the amino acid can dissolve in water. Okay, the hydrophilic amino acid can dissolve in water and form the dipolar ions, and also some of the amino acid also and most of the amino acids have both basic and also acidic group, which known as having the amphoteric characteristic. And then the amino acid also can be divided into 2 large groups, which are the first one is essential amino acid. Essential amino acid are referred to the amino acids that cannot be synthesized by the cell. Okay, however, we can obtain it from our diet, okay, from your foods. Okay, meanwhile, for non-essential amino acid is referred to the amino acid that can be produced by our body, okay, not come from the foods or not come from our diet. Okay, and next is dipeptides. When two monomer, okay, one monomer of amino acid, one amino acid monomer, when they bound together, okay, through the condensation, okay, or removing of the water H2O, they will form a link here that is known as the peptide bond. And now the structure is known as dipeptides. So meaning to say it consists of 2 amino acids linked together by a peptide bond through condensation process. Okay, so the equation will be 1 amino acid combined with another amino acid through the condensation process, thus producing water, and also producing dipeptide. And these equations can be reversed also. Okay, this reaction can be reversed. Dipeptide um, can be hydrolyzed back, okay, or can be break down into back to amino acid, okay, through the process of hydrolysis, meaning you add water to the dipeptide, so it will produce two molecules of amino acid. And they are also a proteins that are, or the enzyme that are responsible to cut this peptide bond, which is known as the peptides. Okay, and then next is a polypeptides. When you have a long chain of amino acid that bonded together through the condensation process, meaning to say we have lots of peptide bonds between one amino acid to another amino acid. Now it will form what we call as a polypeptide. Okay, a long chain of amino acids. And then when two polypeptides, okay, combine together, forming together, they will form a protein. So protein, they have a lots of functions, so many functions, such as it is important for the structure, okay, such as it make up the protein, the collagen protein in our skin, the keratin protein in our hair and also nails, and then it also important for the movement, such as actin and myosin protein in the muscles, and also for defense mechanism, because our antibodies is actually produced from the protein, and then for the storage, such as the zeatin protein in corn seeds, and also important for the signal transductions. Okay, for example, a growth hormone, okay, they make up from the protein, made up from the protein, and also important for catalysis process, in which it made the enzyme. Remember, all enzymes actually come from protein, but not all protein will become the enzyme. So functions of enzyme that to catalyze every chemical reactions in our body, in which this enzyme you will learn later in semester 2. Okay, and then for protein structure, you have to remember there are 4 important levels of protein structure. The first one we call as the primary protein structure. The second one is secondary protein structure. And then we have tertiary protein structure. And then the last but not least is that quaternary protein structure. So primary protein structure is actually referred to a long single chain of amino acid that bonded together by a peptide bond. So we have a peptide bond here, here, until at the end. Okay, and this one is amino acid. For an example, here we have methionine, and then we have valine, isoleucine, proline, phenylalanine, and etc. Okay, examples of the protein that have a primary structure is insulin and also glucagon. And then the next one is secondary protein structure. Secondary protein structure involve hydrogen bonding. Primary only involve peptide bond. However, for secondary protein structure, they involve a peptide bond, and then they involve hydrogen bond. This hydrogen bond, okay, it will cause this long chain to become either folded like this, known as a beta pleated sheet structure, or they will coil and forming the structure that known as the alpha helix structure. Okay, they coil like this. And then next one is that examples of the proteins that have this secondary structure is keratin that you can find in your hair. Okay, next is tertiary protein structure. Tertiary structure is that is referred to the 3D structure of the protein, in which, as you can see over here, it made up from a coil, okay, a coil structure, and then we have a beta plated sheet over here. Okay, and then they will form a structure. Okay, here as you can see, through a several number of through a several number of bonding, such as disulfide bridge, van der Waals interactions, and also hydrogen bonding. Okay, and then when we have two molecules of tertiary protein structure, okay, joined together, this one known as quaternary protein structure, okay, in which it consists consisting of more than one chain of amino acid. Example is hemoglobin. Okay, so this one, as you can see over here, the primary structure, a long chain of amino acid. Example, we have phenylalanine, leucine, serine, cysteine, and so on. Okay, and then the secondary structure. The secondary structure is that we have, this one is the chain, right? Okay, chain of amino acid, and due to the interactions of the backbone of one amino acid to another amino acid through hydrogen bonding, as you can see, hydrogen bonding over here, and it will cause the long chain to give a helix structure, a coil structure that known as a alpha helix. Okay, if like this one, it is known as the beta plated sheet structure. Okay, for the tertiary structure is actually caused by the interactions of R group, okay, from R group of the one amino acid to the second group of the amino acid. Okay, different with secondary structure. Secondary structure is actually interaction between backbone of amino acid to the backbone of another amino acid. Meanwhile, tertiary structure is caused by the interaction between the side chain or the R group of one amino acid to the R group or the side chain of one amino acid. Okay, either by hydrogen bond, disulfide bridge, hydrophobic interactions, or van der Waals interaction, and also can be caused by the ionic bond. Okay, for secondary structure, only involve hydrogen and peptide bond. For primary structure, only involve peptide bond. Okay, and last but not least is quaternary structure, in which consists of two or more polypeptides that are combined together, producing a quaternary structure of the protein. And next, the most important thing about the protein that you have to remember is about the protein denaturation and also protein renaturation. Protein denaturation, we can call it as a as the destructions of the protein, in which you will destroy the protein by what we call that one, by cutting off or by break down the bond, okay, inside the protein. Therefore, denaturation is referred to the process that it will change the shape of the protein, okay, without breaking its peptide bond. It does not break, as you can see over here. This is the shape of the normal protein, and denaturation occur. What will happen? The peptide bond that hold one amino acid to one amino acid is still here. However, the bond other than peptide bonds, such as the hydrogen bond, okay, that involve the interaction between the backbone of amino acid to another backbone of amino acid, and another example is disulfide bridge, that between side chain of amino acid to another amino acid. That one will be destroyed by the denaturation process, and the agent used to destroy it either by pH, okay, very acidic or very basic, high temperature or very low temperature. It might caused by the ionic strength, and also it might caused by the solubility of the protein itself. And so what is renaturation? Renaturation is the process where this long chain of amino acid, they will fold it back to its normal structure. Okay, meaning to say the renaturation process is where the disulfide bridge, the hydrogen bond, the van der Waals interactions, and the ionic bond will form back. Okay, they will form back, thus producing the shape of the normal protein. Okay, so examples of the protein that cannot be renatured back is fried egg. Okay, egg is a protein, right? So when you fry the egg, so what will happen? The egg will be denatured, and once the egg become the fried egg, you cannot renature it back to the normal egg. Okay, cannot. Okay, so this one is a type of the protein that cannot back to its normal structure. Therefore, renaturation does not occur. Okay, the second example is milk. Okay, milk. Okay, when you put it in the freezer, so what will happen to the milk? The milk will become solid. Okay, will become solid at 0 degrees Celsius. However, the shape, okay, when the shape can be back to its normal, okay, when you warm the milk. Therefore, the milk can what we call that one ya? The milk can akan jadi liquid structure. This one is denatured form, but it can renature back to becoming the solid structure when you put it in the fridge. Okay, so that's all for protein. So next is nucleic acid. So nucleic acid is a macromolecules that compose of change of monomer that called as a nucleotides. Therefore, nucleic acid is actually refers to the polymer, or also known as polynucleotide, and the monomer of nucleic acid is nucleotides. Okay, as you can see over here. And there are two classes of nucleic acid. The first one we have DNA, or deoxyribonucleic acid, and the second class is ribonucleic acid, also known as RNA. So the nucleotide structure, which is the monomer of the nucleic acid, okay, they have three main components, which are the first one, it made up from a pentose sugar. Pentose is referred to the sugar that have 5 carbon. So here is carbon number 1, carbon number 2, carbon number 3, carbon number 4, and this one is carbon number 5. Okay, so the carb sugar, either ribose or deoxyribose. Ribose sugar can be found in RNA, and deoxyribose sugar can be found in the structure of DNA. And next is nitrogenous bases. So nitrogenous bases, there are two types of nitrogenous bases. The first one is purine, and the second one is pyrimidine. And next, they also consist of one phosphate group that attach to the carbon number 5 over here. So let's look on further details on each of these three components. So the first component is that the pentose sugar. Okay, here, this one, the pentose sugar, the 5 carbon sugar of nucleotide. So a pentose sugar, as you can see over here at carbon number 2, okay, deoxyribose sugar, they do not have oxygen that attached to hydrogen here at carbon number 2. Meanwhile, for ribose sugar, as you can see over here, there is and oxygen that attach to the hydrogen atom at carbon number 2. So the deoxyribose sugar can be found in the structure of DNA. Okay, that's why the name of the DNA is deoxyribonucleic acid. Okay, due to the deoxyribose sugar. Okay, so as I mentioned earlier, it only have hydrogen atom at carbon number 2 and lack of one oxygen atom. Okay, so differ with ribose. Ribose is the sugar that can be found in the structure of RNA. Okay, so that's why the name of the RNA is ribonucleic acid, because they have here it have a carbon. It contains carbon. It contains ribose sugar, where the ribose sugar have oxygen or the hydroxyl group that attach to carbon number 2. And this one is the normal sugar with one oxygen atom that attached to each of the carbon. So the next component of nucleotide that you have to know is nitrogenous base. So nitrogenous base, there are two types of nitrogenous base. The first one is purine and the second one is pyrimidine. Okay, the difference between purine and pyrimidine is based on their structure. For purine base, they have two large double ring here, in which six member ring they will fuse together with 5 member ring, and then here also 6 member ring fused together with 5 member ring. Okay, examples such as adenine base that you can find in both RNA and DNA, and also guanine base that you can found in both DNA and RNA structure. Meanwhile, pyrimidine, they only have a single 6 member ring over here. Okay, and examples of pyrimidines are cytosine that can be found in both of DNA and RNA, and then we have thymine that can be found only in DNA, and also we have uracil that can be found only in RNA. Okay? Therefore, the nucleotide have one phosphate group, pentose sugar, and also the bases. Okay, pentose sugar either ribose or deoxyribose. If the pentose sugar is ribose, therefore this nucleotide is will make up RNA. If the pentose sugar is deoxyribose, therefore this nucleotide will form DNA structure. Okay. So for the phosphate group, as I mentioned before, it's joined at the 5th. It attached at the carbon number 5 of the pentose sugar. Here is number 1, 2, 3, 4, and 5. Okay, and the nitrogenous bases, ATGC. ATGC is for DNA. Okay, adenine, thymine, cytosine, guanine. They attach at the carbon number 1. For RNA, they have adenine, uracil, cytosine, and guanine. Okay, so they do not have thymine in RNA. Thymine only can be found in DNA. And next, when we have a long chain of nucleotide, now it is called as a polynucleotide, or the polymer of the nucleotides. Okay, so here one nucleotide attached with the second nucleotide, and then the second nucleotide attached with the third nucleotide, and then it will attach to the fourth nucleotide. And the bond that formed between one nucleotide to the next nucleotide is known as phosphodiester bond, that can be formed, of course, through the condensation process. Okay, condensation process by removing the water. So, and there are two types of nucleic acid or polynucleotide. as we already talked about this one. The first one is DNA and the second one is RNA. Okay, DNA, the function of DNA is to carry the genetic information in our body, and it can be found only in the nucleus of the cell. Okay, and the structure of DNA is a double helix structure because they have double strand. This is the first strand and this one is the second strand. And then the first strand, they will bond it together with the second strand through hydrogen bond between the amino acids, between the nitrogenous bases of this strand to nitrogenous bases of the nucleotides of the second strand. Okay. As you can see here, adenine form two hydrogen bond with thymine. Okay. Purine with pyrimidine, always pair purine with pyrimidine, and then guanine form three hydrogen bond with cytosine. Okay, this one also purine with pyrimidine. And of course, the sugar that can be found in DNA is deoxyribose sugar. Remember, deoxyribose sugar is the sugar where they do not have oxygen that attach to carbon number 2 of the sugar. And then the pentose sugar, the nitrogenous bases that can be found only in DNA, are thymine, adenine, cytosine, and also guanine. And adenine always bonded with thymine, okay, by two hydrogen bond. Meanwhile, cytosine always only pair with guanine, okay, by three hydrogen bonds. Next is RNA, okay. As you can see over here, RNA is important for protein synthesis and can be found mainly in cytoplasm, but also can be found in, some of them can be found in nucleus, okay, and their strands are quite short compared to DNA, and they have only one strand only. They do not have a single strand, okay, and the sugar is ribose sugar. Remember, ribose sugar is a pentose sugar where they have hydroxyl group OH that attach to carbon number 2 of the pentose sugar. And the nitrogenous bases that can be found only in the RNA are adenine, uracil, cytosine, and also guanine. And there are three types of RNA. We have messenger RNA or mRNA, ribosomal RNA or rRNA, and we have transport RNA, also known as tRNA. Okay, now let's look on the complementary base pair. Complementary base pair is the pairing between one nitrogenous base of nucleotide with the nitrogenous base of the second nucleotide. So as you can see here, adenine only, okay, bonded with thymine in DNA, okay. If in RNA, they only bonded, this one they only bonded with thymine and they forming two hydrogen bond. Meanwhile, guanine always and only bonded with cytosine, forming three hydrogen bond. Okay, so you can see over here. Therefore, the structure is like this. Okay, spiral. This one we call as a double helix structure. Okay, now back to the types of RNA. Messenger RNA function is to carry genetic information from DNA to ribosome for protein synthesis. Ribosomal RNA is the one that used to make up the ribosome. For transport RNA is that function to bring the amino acid to the ribosome for protein synthesis. All of this RNA you can learn, and you will learn about it later when you are in semester 2. Okay, so in conclusion, so what is the difference between DNA and RNA? DNA type of sugar is deoxyribose, RNA ribose sugar. Number of strand, DNA have 2 strand, double helix structure. Meanwhile, RNA have only a single strand, which is known as single helix structure. And then nitrogenous bases that can be found in DNA are adenine, cytosine, guanine, and thymine. Thymine only can be found in DNA. Meanwhile, in RNA, you can find adenine, cytosine, and guanine as a nitrogenous bases, and uracil also that only can be found in RNA. And then DNA only can be found in nucleus. Meanwhile, RNA only can be found in some of them in nucleus, but most of them can be found in cytoplasm. Okay, so that's all for biological molecules. So therefore, summary of the organic compounds or the biological molecules is that carbon with hydrogen, they will form a hydrocarbon, and then hydrogen with carbon with oxygen, they will form carbohydrates and also lipids, and then hydrogen, carbon, oxygen, and nitrogen, they will form amino acid and also protein. And hydrogen, carbon, oxygen, nitrogen, and sulfur, they can form amino acids, more amino acid, and also protein. And then we have hydrogen, carbon, oxygen, nitrogen, sulfur, and also phosphate, which is the phosphorus. Okay, they can form the nucleic acid, which are DNA and also RNA. So this one also you have to remember. Okay, what is the monomer of carbohydrate? Monomer of carbohydrate is monosaccharide. Monomer of lipids have no monomer, but they have a smaller component that known as a fatty acid. Monomer of protein is amino acid, and monomer of nucleic acid is nucleotide. And the polymer of carbohydrate is polysaccharide. Lipids do not have polymer, but they do produce form the larger molecule that known as triacylglycerol or triglyceride. And then the polymer of the protein is polypeptides, and the polymer of the nucleic acid is polynucleotides. And the linkage between monomer of carbohydrate is glycosidic linkage, the bond, and the bond between the smaller components of the lipids is ester linkage. And then the bond between the monomers of the protein is peptide bond, and the bond that form between the monomer of the nucleic acid is known as phosphoester linkage. Okay, so this one you can read by yourself later. So I think that's all for today, class. Thank you.